Yes, welcome again to the Base to Base Biotech podcast, and this is number 72, and it's going live on August the 21st. This week, you could not make up the fact that 5 straight days of rain has coincided with an electrical fault in my car, meaning the windows won't go up. Still, it's now nice and clean inside and out, and I can wash my feet as I drive. School is back here, so the house is very quiet, and I can't say that I like it too much. And that's about it for what's happening. I'm sure I'll remember something amusing or interesting that happened this week as soon as I finish recording. I do think the summer vacations are drawing to a close because the inbox is growing again. Most of it is irrelevant though. I've also had lots of bank invoices that have been paid by companies I've never heard of, to a range of banks across the world that I don't have accounts with. I suspect it's probably spam. Last time we had a conversation with JJP Biologics about specially built antibodies to tackle autoimmune diseases and cancer. This week it was remarkable that the interview worked so well given we had 3 guests and 2 other people watching, and me. The screen looked like the Celebrity Squares show. I don't think that still exists in the UK, but I think it's called Hollywood Squares in the US, and it might still be running as far as I'm aware. What this has to do with biotech, I have no idea. I sidetrack very easily. Oh yes, there were lots of people on the call. We chatted about the Flexible Automation for Scalable Health program, also known as FLASH, and it is focused on the development of scalable cell-free DNA bioproduction capabilities. So, our guests are John Schiel, Program Manager at ARPA-H, DNA Script co-founder and CTO Xavier Godron, and John Nelson, Senior Biosciences Principal and Flash Program Lead at GE Healthcare's Healthcare Technology and Innovation Center. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, or sponsoring a podcast episode, or for event coverage, or maybe just to tell me your vehicle horror stories, so I don't feel so bad. Right, let's first run through some of the news headlines from the past week from articles at deeptechdigest.com, just in case you missed them. It's still a little bit light on news, but I think that will start to pick up very soon. Tagomics introduced a DNA-based blood test for multi-organ drug safety monitoring. Jazz Pharmaceuticals is acquiring Actio Biosciences to expand its rare epilepsy portfolio, and IASO has acquired cell therapy company Medisix. Researchers uncovered a new driver of immunotherapy resistance in pancreatic cancer. An RSV antiviral drug has received FDA approval for a phase 2 trial, and KTU chemists in Lithuania have developed dual-action compounds to fight cancer. And you can read all of these and plenty more at deeptechdigest.com. Alright, let's get to this week's conversation, and this week it is with 3 people in different places and no technical issues, which was a miracle. DNA Script and Prime awardee GE Healthcare have been awarded up to $26 million in funding for a 4-year initiative from the Advanced Research Projects Agency for Health, which fortunately is shortened to ARPA-H. The award will support the advancement of the FLASH program, which is focused on the development of scalable cell-free DNA bioproduction capabilities. So, we'll hear about all of these things and more. Of course, the project is a research project that's just starting, so the program will likely need to go through some regulatory steps before it's ready for commercialization, but hopefully you'll get a clear picture of what's going on in this chat with John Schiel, Program Manager at ARPA-H, DNA Script co-founder and CTO Xavier Godron, and John Nelson, Senior Biosciences Principal and Flash Program Lead at GE Healthcare. All right, thanks so much to all of you for being with us today. Maybe first we could just do a few brief introductions.
I'm John Schiel. Before coming to ARPA-H, I was with the National Institute of Standards and Technology. I was part of their biomanufacturing program for about 15 years. So I ran an analytical lab there where we were developing reference standards for different therapeutic products and performing analytical characterization in collaboration with the biomanufacturing industry. Near the end of my term at NIST, I was writing several documents and kind of thinking about the future of biotechnology in the biopharma industry, and had an opportunity to come to ARPA-H to run some of the bold mission-driven research projects that ARPA-H is working to stand up.
I'm John Nelson. I'm a senior principal scientist at GE Healthcare. I've been here essentially since 1997 when it was Amersham, and then it became Amersham Pharmacy of Biotech, and then it became Amersham Biosciences, and then it became GE Healthcare, and then it became GE Global Research, and then GE split up, and then it's GE Healthcare. So it's the same job for the last 28, 29 years now. I'm a molecular biologist, and I develop molecular biology tools, and I'm the PI for the FLASH program.
My name is Xavier Godron. I'm one of the founders and CTO of DNAscript, a company which we created with my partners in 2015. We have developed a new way to synthesize DNA. So we go from digital sequence to making DNA molecules, and we do that enzymatically, which is basically the way nature makes DNA, which is a contrast to the current chemical DNA synthesis that is used by most of the industry. And as we progress in development of our technology, we are moving towards assembling longer pieces of DNA together and to get towards bioproduction. And so we've been working on that with the GE team for quite some time. I think we started in 2020, 2019 maybe. And with this new ARPA project, we'll be able to push that forward even more.
All right, so I guess this one's for John specifically. I wonder if you could tell me about ARPA-H and how it's helped others.
Absolutely. So ARPA-H stands for those that may not know that are listening, the Advanced Research Projects Agency for Health. And I'll talk a little bit more about this, but we're a relatively new organization within the United States government. We're a separate organization within the Department of Health and Human Services. So we report directly to the Department of Health and Human Services. Our role is really to be a non-traditional funder. So we look for very bold, high-impact research efforts with the goal of transforming health outcomes. So we really want to bring technologies that maybe would take decades to develop and bring them into the year's time frame so that we can get innovations to biomanufacturers, to patients, to clinicians faster than they would under traditional funding mechanisms. So with that, we do really, again, try to enable the next generation of technologies. And Flash is a specific, very excellent example of that, where we're looking to develop automated DNA manufacturing technology that was catalyzed by Xavier, John, and their teams at DNAscript and GE Healthcare, and looking to really get that into the hands of researchers. So you asked a little bit about how we help industry, academia, innovators throughout the ecosystem. And I'll say we're a very young agency. So I was actually the first program manager in Scalable Solutions, which is the office that we sit in. So a little over a year and a half, I was brought on over on detail, and we essentially stood up this new office, Scalable Solutions. So we're very, very early, but we have several programs launched, a lot of funding at industries that are doing revolutionary things in health intervention. So again, this isn't just biomanufacturing and biotechnology, but health interventions across the landscape of medical care. And we're seeing really strong commercialization signals through several of our efforts. So we really look, again, to take those demonstration products and bring them to commercialization. So certain success pathways could be moving through regulatory hurdles and pathways, getting follow-on capital, but really moving things to commercialization, whether that be a manufacturing capability or a therapeutic product.
And so specifically, what is Flash?
So the FLASH program is being designed to create DNA constructs, DNA expression constructs, genetic medicines from scratch. So it's a platform that you can send a text file to, and some number of days later, potentially as short as 3 days later, your nucleic acid comes out the other end. So it's a completely automated platform that you just send the instructions and oligonucleotides are printed, they're assembled, constructs are made, they're amplified, they're purified, and it comes out the back end ready to use.
EDS technology and syntax, we've got a lot of acronyms.
Yes, so EDS technology and syntax, this is the first block of this flash system. What EDS does, it means enzymatic DNA synthesis. So it's the new technology that I mentioned in my introduction, where we're using enzymes to add nucleotides one by one to synthesize the sequence that we want. And syntax is the machine that automates that. So the syntax machine, it will take a file of a few hundreds of different digital sequences of DNA. Each of them can be a few hundreds of nucleotides in length. And the machine will print them in plates. In one of the well of the plate, we'll have one oligonucleotide. And that's the output of syntax. So syntax, we call it a DNA printer sometimes internally. So it's a platform that is commercially available today. But what's interesting in this Flash program is then we're not stopping there. We're not stopping with smallish oligos. We are taking this plate of oligos and assembling it into full genes of multiple kilobases of length. And we are also amplifying those genes to have enough material so that it can be usable for a lot of downstream applications. It can be used for mRNA, can be used for CAR-T therapies, for AAV, antibody development, etc. And we think that this full tech stack from digital sequence to amplify DNA will bring value into accelerating drug development. And also we want to use that for bioproduction to get DNA produced faster, closer to the patient, accelerate clinical development and treatment in the case of personalized medicine.
And so what is the funding and what does that mean for these programs?
Yeah, so the funding, ARPA-H has committed up to 26 million over this 4-year initiative with GE Healthcare and DNA Script to essentially build the Flash system. And then we want to deploy this as well to early adopters of this technology. So at ARPA-H, we like to emphasize, this is very unlike traditional, seed funding rounds or grant funding. So this isn't an experiment. It's a runway to actually take this technology that's been de-risked already by these partners and bring it to commercial success. So we're building an automated deployable, so this is potentially a distributed manufacturing platform that any institution with the relevant expertise could actually adopt for their genetic medicine manufacturing. So we're funding, again, this development and distribution. I think definitely taking into account the vision of what this has the potential to really catalyze. It really is the ability to make your own DNA. So there's a bright, bright future, and we can talk a little bit more about how this kind of parallels other technologies that have translated in this field, some of the big impacts that I think it can have.
And I should note that this project is currently in the first year of its 4-year program, so it's fairly early on in the program.
And how is the partnership going to operate?
GE is the prime on the program, so I'm the PI for the program. And this is a continuation of an earlier program, which was funded by DARPA, where we collaborated with DNA Script to really de-risk a lot of the aspects of this that were less well known back then. And at the termination of that DARPA program, then we started this ARPA program, and we're continuing on with the same team that had been working since, I think that proposal for the DARPA program was submitted right at the very start of COVID. So January of 2020 was when this whole thing started off. But at this point, it's GE Healthcare. We provide the DNA amplification technology, which is in the workflow. DNA Script prints the oligos, does the assembly reaction. The team also has Johns Hopkins University. Winston Timp's lab is helping us with the bioinformatics. It's, I think, important to mention that the DNA coming out of this is very high fidelity. There are strict requirements on the error rate that we're allowed to have. And since the program doesn't involve a clonal selection of an individual DNA molecule, it's all done in bulk reactions. There's no way to take a single molecule, amplify it, sequence it, make sure it's perfect. What you make at the beginning has to have high enough fidelity so that it reaches that goal at the end. So one of the difficulties that we've had is there's actually no DNA sequencing platform that can sequence at high enough fidelity on standard runs to determine what the error rate of our DNA is, because it is such a low error rate. So we've brought on Johns Hopkins. They're experts in DNA sequencing technologies and bioinformatics. And with them, we're developing a workflow to develop DNA sequencing methods that can prove that the DNA we're making is extremely high fidelity.
Looking at the field in general, is DNA synthesis a problem or a bottleneck? And if so, what issues is that causing?
Yes, DNA synthesis is a bottleneck in many, many different fields. One of the challenges is in my opinion, it's too centralized. So it makes it slow, it makes it dependent on supply chain. It makes people lose control over the processes because they always need to order DNA outside. And the specifics of what we're trying to achieve here in this program, which is really a cornerstone towards bioproduction, DNA synthesis for use for therapeutics relies on bacteria for two very important steps in the process. The first one is selection of a perfect DNA molecule, because the current ways to make DNA are quite error-prone, and we use bacteria to select a perfect molecule. And the second one is amplifying this perfect molecule to have enough quantity, enough mass, so that it can be usable in the application. And the fact that we use bacteria for those two steps puts a lot of complexity in the production process. You need to operate very complex processes in clean room. You need to invest a lot in documentation. You need to have a master cell bank of your cells with the DNA, working cell bank, etc. And that whole process, the consequence of that is that having GMP DNA today, it costs hundreds of thousands of dollars for small batches, and it takes 3 months to get the DNA. And that's a problem because people doing drug development, they want to move fast and they want to try different constructs sometimes. One extreme is people doing personalized medicine, where one patient needs one DNA batch, and you can see the economics and the timeline are not working for that. If you need to spend 100,000 for the first step of your process, it's going to be challenging to commercialize a successful product. So that creates a whole bottleneck there for this type of company, and that becomes a big challenge for those companies working in personalized medicine. So our approach here, basically, at DNScript, we are solving the first aspect of what bacteria does, meaning we want to provide very high fidelity DNA without bacteria in a process that could be easy to do for production. And G is solving the second part, which is making amplification at high purity completely in vitro without having to rely on the dirty bacterial process that's used today.
We had Base Bio on the podcast recently talking about what they're doing with synthetic DNA. Is there a big field here? I mean, are many companies working on this, and is it making a difference?
There are a few companies working in cell-free amplification, which is the focus of Base Bio. We could mention, for instance, Touchlight or Singoy, who are moving towards GMP with this. Actually, the history of the technology behind all of those companies is rolling cycle amplification, which you have one of the esteemed inventor on this call, John Nelson. The first kits for rolling cycle amplification have been developed and commercialized by GE and now Cytiva, I believe, but John could comment more on that. As for your question, whether it makes a difference, it does make a big difference, because one of the super important new thing that happened is that this technology is entering the clinics, with a few of those companies having been approved to enter early phases of clinical development for different therapeutic applications. And that makes a huge difference, because the full potential of the technology is when every one of those different companies and technology can be in the clinics in humans and can have an impact on the rollout of new drugs into the market and provide better care to patients.
You can almost think of it like the photography. It used to be that you had to take pictures, send it away, have all the high-tech stuff done for you, and then you get back prints. And these days, it's all in the hands of the users. And our aim is to essentially do the same thing with this technology.
And how can enzymatic DNA synthesis impact future research and therapeutic development?
I think the headline benefit really is speed and control. So instead of waiting on an external vendor, this team is building the capability that you can synthesize high fidelity DNA right in-house and iterate over a period of days on the DNA constructs that you want to test for your therapeutic development. So if we correlate this to next generation sequencing technology and the Human Genome Project, the rate of acceleration of that technology was very, very fast, and the uptake was fast. And so researchers across the world now can either perform next-gen sequencing in their own lab or send it off to core facilities that are regional and get results very quickly on whole genome sequencing, patient genome sequencing. And that's really advanced biomarker discovery, clinical diagnostics. Every field that we can think of that NGS touches has advanced with that technology. And here we're talking about, now let's put the ability to read DNA. Now let's create the ability to write DNA. and think about what we can do to iterating on discovery pipelines. So it's much faster to iterate different DNA constructs, look for new therapeutic proteins, and I think that will accelerate the development of new personalized neoantigen cancer vaccines, gene editing approaches, in vivo genetic medicines. There's so many different therapeutic avenues that can be tackled by this type of technology that I think there's a super bright future for advancing discovery and development in several phases.
As part of the program, we've done a lot of voice of customer, and every time we contact a new core lab, a new PI, professor, somebody out there that is working actively on making different types of genetic-based therapies, they all say that the future is being able to do it themselves and not have to phone a clone all the time. And some pharma companies hesitate to send out their sequences because there's proprietary things in them. They like to be able to have control over what they're making. So speed, the ability to just decide, I want to do this experiment, go to the machine, send your stuff in it, wait a couple days, and you're ready to go. That's a game changer for these labs.
And how does it actually work, the process?
We've got a multi-instrument platform that you send the digital sequence to the platform that you want to have made. The platform prints out the oligos with the syntax machine. Those are then transferred into a liquid handler that then assembles the oligos into blocks, gene blocks. The gene blocks are then circularized and dropped into a rolling circle amplification reaction where the DNA is bulked up. I call the RCA reaction a DNA printing press. It's a circular piece of DNA, and it's copied around and around by a DNA polymerase that's very high fidelity. And you end up with long pieces of DNA that contain your instructions repeated over and over and over again. That DNA is then purified and delivered in a tube to the end user, all right on that one benchtop platform.
You mentioned some of the positive aspects of, not having to send out proprietary information and things like that. Are there any other positives for companies working in this space?
Well, everything in the system is endotoxin free. So we know that the DNA coming out the back end is highly purified and is capable of being used in a variety of different end uses. We've found that our amplified DNA can be used in place of supercoiled plasmids in essentially every possible way that supercoiled plasmids have been used in the past.
Ultimately, when it comes right down to the very end of this process, not necessarily now, but what are the implications for patients at the end of the line?
Generally, for patients, it's good that R&D in drug development in pharma has good tools because that will enable those companies to put in the market new drugs on a faster pace. I think I want to insist a little bit on one space that I think patients would benefit the most. Again, it's personalized medicine. For any type of personalized therapy, it is very important that the DNA can be produced at high quality, at a decent price, so that the therapy can touch the whole base of people that would benefit from it, and also at a fast pace. This is the one indication in which the time, we call it sometimes the needle to needle time in oncology. The first needle is the moment when you will take a biopsy of the tumor and sequence the tumor to identify the mutation of the cell. And then from that, you will design a personalized drug. And this drug will teach your immune system to recognize the cancer cell as a bad cell. And it will teach your immune system to destroy it, basically. And in order to do that, you need to inject the drug at some point. So that's the second needle time. And the shorter this time frame is, the better is the outcome for the patient. That has been widely published in the literature. If you can administrate the personalized drug faster, it will mean better outcome for the patient. And I think this is where the difference is the most meaningful, because you not only enable R&D and bioproduction to be faster, to be cheaper, to have more control, more confidentiality, we would actually change the outcome on the patient by being able to produce the drug quickly. And to me, that's the most exciting thing, right? To have an impact on something like that. It would be industry shaping, but also would change some patient life.
And tacking onto that, I think Xavier is correct. Personalized medicine is the way of the future, and this technology is the way to get there. We talk about often in scalable solutions, taking, the dirt roads in the country and turning them into thoroughfares and getting there quickly. Personalized medicine is going to need a personalized manufacturing solution. And I think that's what this team is offering. So many of the advantages that John and Xavier spoke about, no endotoxin burden, rapid synthesis, and being able to custom make the DNA for a patient, those are all the unlocks for this field of personalized medicine, and this team is really developing those.
Are there other applications beyond personalized medicine?
We have certainly, since 2020, when we started working with DNA Script, have collaborated with a variety of people that are working on screening through things. And so being able to make a construct quickly allows you to reiterate. And so if you're doing drug development or enzyme development, antibody development, and you want to make a version, test it, make a little change to it, make it again, test it. If you've got to wait a month to make it every single time, 6 iterations is half a year, whereas if you're waiting just a few days, 6 iterations is just a couple weeks. So certainly in that context. But then if you just think about where DNA is used, there are tons of other places that DNA is used that aren't actually drugs, but are certainly useful research uses of the platform.
There's a bit of a new trend coming with all the AI protein design, where what John described that sometimes we call design, build, test cycle. So designing a new construct, building it, and testing it. This loop, we want to be able to iterate as fast as possible to optimize a new solution. And yes, there is this new trend of having AI doing the design. And so the job is really to be able to have good operations to cycle through the loop as fast as possible. And then when we have a good design, lock it up and go towards the clinical development. This is usable for protein design, but it's usable as well for CAR-T therapies, which is kind of the same in some ways, and a wide range of different applications.
I was going to ask about AI. How is that impacting on this whole process?
I think it puts some pressure on the DNA synthesis industry to go faster and go high quality. I also hope it pushes a bit for decentralization so that people control really their loops. There's this other big trend that people call lab in a loop, where people want to have all the automation to make this design, build, test cycle happen internally. And that's the differentiation between one company doing drug development and the other is the ability to have this whole process run faster and faster. So yes, I think it's good for the industry. I think it will lead to better drugs. And I think it puts also more of a bottleneck on the DNA part, on the build part, because it really helps the design.
Are there any challenges associated with this?
Tons of challenges. There are challenges, it's very new, so people are still figuring it out. One challenge that I want to mention as well is biosafety, and that's one of the important part of the program is ensure that we have control over what is being synthesized and that we can control it. But AI is making design a lot easier. We've all heard about the guardrails that the AI companies are putting on biology. Anthropic, they are being very careful in the way that they deploy that. We also want to be careful in the build part of the process.
And for the program itself, what are the timelines associated with that? You mentioned it's a 4-year program.
So in the first year, we are rushing towards instrument development, and this team has been all hands on deck to complete a prototype system that can carry out everything, as John mentioned, from tech sequence in to purified DNA product out, and looking at many different categories of DNA. We then intend to, in the following 3 years, identify and adopt early adopters that can beta test this equipment in their own labs and generate new medicines, new therapies, really test the technology, but also feed back to these developers to continue to iterate and build on the platform that they've already built. So it's a 4-year timeline overall, where we'll get both development and product evolution again towards that highly commercializable and robust instrument at the end. And kind of tagging on a little bit to what Xavier said, the biosecurity has been at the forefront of our minds in this effort. We're actually partnering with Los Alamos National Laboratories as well as part of this effort to ensure that we're developing all of the necessary safeguards in place, that this really will be the pinnacle of DNA manufacturing, but also the pinnacle of safe and secure printing of these sequences.
And at the end of the day, what does success look like from this program?
I think it's different for all stakeholders, right? So I think for patients, it's probably the most important, and let's start by that. It's being treated in the best possible fashion. For our customers, in the end, it's going to be to have access to good quality DNA fast, be able to execute better on their clinical development. for us at DNAscript, and I'm sure for GE, is to build a solution that can be successful on the markets and help both companies to be more commercially successful, and as well execute on our mission to make biology more programmable and helpful for healthcare.
When you take a job for a medical technology company like GE Healthcare, one of the things you hope is that you can develop a product that is going to save lives, is going to make people's lives more healthy. And I think this program really has the ability to do that. So it's been a very personally fulfilling program for me to be involved with.
Yeah, and I think the beauty of working with this team is we all have such a shared vision for this DNA. So having this capability, again, we're creating a capability, not a product in this program. And so when people talk about catalyzing an ecosystem, this technology really can catalyze an ecosystem. Anyone can now, with the proper expertise and experience, could begin a therapeutic development program. And I think the rapid iteration advantages of this platform, better health outcomes for everyone, really is the potential that this capability has. And so I think the patients are always at the forefront of our mind, but by providing this capability to developers in industry, academic medical labs, core facilities, will make their jobs a lot easier so they can also benefit the patients that they're trying to benefit faster. So really a lot of advantages across the entire development life cycle.
And I assume it has implications and applications beyond just the US as well.
Absolutely. So I think, we're really approaching biotech as an agency. That friendshoring or partnership with our allied countries is extremely important to make sure that we're developing the best technology to get to patients. And that's one of the reasons that ARPA-H can fund internationally, and we have international partners in these programs, is that we're really looking forward to collaborating with the best and the brightest in this field to bring these solutions to patients.
Are you familiar with neoantigen cancer immunotherapies? That is, to me, I think, one of the most exciting possibilities here. That technology is really just developing now. There's many, many clinical trials going on. And it was really, I think, that whole field of neoantigen cancer therapies was initiated because whole genome sequencing became so inexpensive that it allowed you to take a biopsy and sequence the whole exome of that cancer and find all the mutations that were in it. And, so there were professors and academics all over that were saying, once we can sequence the cancer, we can find out how it's different from the rest of the body and develop an immunotherapy against that. And so what we're seeing is since 10 years ago, it became possible to inexpensively sequence an exome. Takes about 10 years to write that NIH grant proposal, get it accepted, start your program, do the clinical trials, get 3 years worth of results. And so what we're seeing now is we're seeing dribs and drabs of these first publications coming out with people that have breast cancer, colon cancer, pancreatic cancer that are alive that prior to the clinical trial would not have made it. And so seeing those kinds of publications and where that person, when they have that cancer, that biopsy is taken, a timer has gone off. And getting them the therapy as quickly as possible is the number one goal. And so I think John Schiel, he came to us when we were doing the DARPA program and said, hey, I see a need here for a technology that does this. Would you guys be able to develop a platform that could just spit these things out? And Xavier and I said, yep, absolutely.
Yeah, and there was one study actually that looked at personalization of sort of the previous generation of cancer therapies. So radiological therapy, small molecule antibodies, and they increased median-free survival by 90% by personalizing existing approaches. And that's not even training the immune system. So in this case, we're taking personalized neoantigen therapies, training a patient's own immune system to attack their own cancer. I think we'll surpass that 90% by miles with this kind of technology. And it simply isn't scalable without this rapid modular DNA printing because we have to make one batch, one patient. And we can't do that with these large scale centralized processes. This is really the way of the future. Pretty exciting.
Well, that's definitely an impressive program, and as you heard, major potential for positive impacts for patients down the road. And I'm sure those 4 years will just fly by. I'll definitely be interested to keep up with their news over that time. And so that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please do feel free to get in touch at jim@deeptechdigest.com. And so until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.